Welcome back again in a new video session from Biology Boost Up, and in this video tutorial session you will get the whole concept regarding RNA or ribonucleic acid. So now let's start. The ribonucleic acid is a single-stranded polyribonucleotide which functions as a carrier of genetic informations from DNA to cytoplasm and takes part in protein and enzyme synthesis. It is non-hereditary nucleic acid except in some viruses. At places, RNA may appear partially double-stranded due to folding or coiling of single strand. Double-stranded RNA can be seen in viruses only. The axis or backbone of RNA is formed of alternate residues of phosphate and ribose sugar. Phosphate combines with 5' carbon of its sugar and 3' of next sugar, similar to the arrangement found in DNA strand. The nitrogen base are attached to sugar at 1' carbon of the latter and are of 4 types: adenine, guanine, cytosine and uracil. Now the types of RNA. Basically the ribonucleic acid are two types, that is genetic RNA and non-genetic RNA. The genetic RNA again subdivided into two categories, that is double-stranded genetic RNA and single-stranded genetic RNA. Whereas the non-genetic RNA are many types like mRNA, t-RNA, r-RNA and others like antisense RNA, hnRNA, snRNA and scRNA. The genetic RNA helps to carry the genetic material from the previous generation to the next generation, whereas the non-genetic RNA play an important role during the protein synthesis. Now let's talk about the structure of different RNAs. First, the structure of mRNA. Now look, the first point is cap region. At the 5' end of mRNA, there are some nitrogen base containing methyl groups. This region is called cap region. Next of the cap region is non-coding region 1. It is the next part of cap region is known as non-coding region 1 or NC1. This is 10 to 100 nucleotide long region, and this region not helps during protein synthesis. Next, the initiation codon. The AUG forms the initiation codon region, which is the previous part of coding region. Next, the coding region. This region is made up of by about 1500 nucleotides, and this region takes a part during protein synthesis by translation. Next, the termination codon. Next part of the coding region is the termination codon region. The codon of this region may be UAA or UAG or UGA. And on the termination codon region, the translation process become totally stop. Next, the non-coding region 2. This is the next part of the termination codon region, which is about 50 to 150 nucleotide long. And the last part of mRNA is poly A tail region. At this 3' end of mRNA, there are 200 to 250 adenine-rich nucleotide present, and those forms the poly A tail region of mRNA. Now next, let's talk about the tRNA. The tRNA, also named as sRNA or soluble RNA or supernatant RNA and adapter RNA. 10 to 15% of total cellular RNA is tRNA. It is a small molecule with about 74 to 95 or 94 ribonucleotides long. Sedimentation constant is 3.8 S. tRNA are made up of a single-stranded polynucleotide chain. The unique features of tRNA. In addition to usual nitrogen base adenine, uracil, guanine and cytosine, tRNA contains numbers of unusual bases. Those unusual bases protect the tRNA molecule from dehydration by RNase when the tRNA are floating freely in the cytoplasm. The structure of tRNA. First, the amino acid acceptor arm. Double helical, both 5' and 3' ends of tRNA, 7 base paired, unpaired. At the 3' end, 5' CCA 3' protrudes with OH at the tip. Next, the site of attachment of amino acid. The COOH or carboxyl group of specific amino acid joins with hydroxyl of adenine in CCA to form amino acyl tRNA. Next, the D-arm. DHU or D-arm consists of stem and loop with unusual pyrimidine nucleotide, that is dihydrouracil. 4 base pair stem with a loop contain dihydrouracil. Recognition site for the specific enzyme amino acyl tRNA synthetase that activate the amino acid play an important role in the stabilizations of the tRNA's tertiary structure. Next, the T-phi-C arm. This name for the presence of sequence T-phi-C, thymine pseudouridine cytosine, where pseudouridine is unusual base. This arm also consists of stem and loop. Stem consists 5 base pair. Outermost of those pair is GC. Loop consists of 7 unpaired nucleotides. This loop consists a ribosome recognition site. Now next, the Anticodon Site or arm. This arm also consists of a stem and loop. Stem consists of 5 base pair and loop consists of 7 unpaired nucleotides. Out of 7 unpaired nucleotides, the middle 3 form anticodon. Anticodon recognize and codon of mRNA and bind with its. Now the structure of rRNA. rRNA is single-stranded, made up of many nucleotides. It is linear, or sometimes it forms bonds with adjacent complementary bases. In the rRNA, the adenine attached with uracil by 2 hydrogen bonds and the guanine attached with cytosine by 3 hydrogen bonds. As there are no complementary nitrogen base are present on the linear part of rRNA, hence the ratio of purine and pyrimidine are not equal. Out of total RNA, 40% is rRNA. This rRNA can be classified into 3 categories. First, high molecular weight, that is more than 1 millions. Second is higher molecular weight, that is less than 1 lakhs or 1 million. And at the last, the low molecular weight rRNA.